Howdy, this is Lab Report 10 PCR, Group 2, Ana Martinez, and
Hannah Garcia, Microbiology Fall 2024.
Introduction and Significance. The purpose of this lab is to use PCR polymerase chain reaction to identify the presence of E. coli gene in the food samples. Since E. coli is a common bacteria associated with foodborne diseases, its detection is significant when assessing food safety and preventing potential outbreaks in society. The materials. These are the materials for the lab. Methods: PCR setup. Label the PCR tubes according to the samples to be tested. Example: food sample 1, which is A; food sample 2, which is B. Positive control and negative control. Prepare the PCR master mix by combining the following components for each reaction. Add 8 milliliters of the forwards primer and 24 milliliters of the reverse primer to each PCR tube. Add 1 microliter of DNA template extracted from food samples to the corresponding PCR tubes. Add 22 microliters of the PCR master mix to each PCR tube. Vortex the PCR tubes gently to ensure proper mixing. 2. PCR amplification. Place the PCR tubes into the PCR thermocycler. Run the following PCR program. Initial denaturation 94 degrees Celsius for 3 minutes. Denaturation 94 degrees Celsius for 3 seconds. Annealing 57 degrees Celsius for 30 seconds. Extension 72 degrees Celsius for 30 seconds. Number of cycles 35, and the final extension 72 degrees Celsius for 60 seconds. 3. Gel electrophoresis. Prepare 1.5% agarose gel using TAE, tris acetate EDTA buffer. Add gel red RM or other DNA stain to the gel. Load the PCR products 20 milliliters into the wells of the agarose gel, along with a DNA ladder marker for size reference. Run the gel at 150 volts for about 30 minutes until the bands are sufficiently separated. 4. Visualization and analysis. Visualize the gel using a gel documentation system under UV light. Analyze the PCR results to determine the presence or absence of the E. coli gene in the food samples. Compare the results to the positive and negative controls. Interpretation. If the PCR products show bands of the expected size, it indicates the presence of the E. coli gene in the food sample, suggesting a potential food safety concern. If the PCR products show no bands, it indicates the absence of the E. coli gene in the food samples, which is reassuring for food safety. 6. Food safety assessment. Based on the PCR results, assess the food safety risks associated with the presence of E. coli in the food samples. Take appropriate measures to ensure the safety of the food and prevent potential foodborne disease. Results and observations. The following slides are the results and our observations of the gel using a gel documentation system under UV light. The pictures are our results and the observations are based on what we recorded on our lab notebooks.
Results and observations. DNA ladder. Molecular weight fragment to estimate the size of DNA fragments for reference. A. Visual bands of similar sizes control positive, suggesting the presence of E. coli gene. B. Visible bands of similar size as control positive, suggesting the presence of E. coli gene. P. Positive control. Visible bands are present, indicating the effectiveness of the PCR in amplifying the E. coli gene. And negative control. Visible bands are present, indicating a possible error since it might suggest the presence of E. coli or the use of TAE buffer. Conclusion. Although both of the samples and the controls, positive and negative controls, had the same visible bands, the negative control should have showed no bands. This error in the negative control could have been because of possible contamination during the experiment. The use of TAE buffer instead of nuclease-free water for the gel preparation, since it may contain traces of nucleic acids or impurities that serve as unintended templates during PCR, leading to false positive results in the negative control. This error highlights the importance of using high purity reagents and ensuring correct aseptic techniques during PCR experiments. Key measures to ensure safety of food include washing hands, properly cooking food, and storing food. Thank you.